Hello and welcome to the Cheeky Science Show, where today you're going to get cultured in cell culture. So to be more specific, this video will look at some tips that are useful for trainers or beginners in cell culture, and it will be more about good practice as opposed to technique, since techniques in cell culture can only really be done in person, and my drawing talent is, just not good enough to sketch it out for you. And a part of this channel really is dedicated to me sharing lessons that I've learned from others in an attempt to help to try and remember these lessons myself, but also to spread this knowledge that obviously could be useful to you as well. And this video is also to try and explain the mysterious concept of what someone is actually doing when they say they're splitting their cells or changing media without getting strange looks from friends. Okay then, so what actually is cell culture? Well, according to the definition that I found, cell culture is the process by which cells are grown under controlled conditions, which generally means outside of their natural environment. So it's the ability to maintain cells of interest that may have been isolated from actual living tissue under carefully controlled conditions. And there's actually a very detailed Wikipedia page that goes into so much detail. It's all you could ever want to know about cell culture. So, as promised, here are some tips to get you cultured in cell culture. So the first tip is how to freeze down your cells tactically. So the thing with cells is that they grow and then they divide. And so if you start with a certain amount of cells to begin with, you're going to have more cells at the end. And so the excess cells can be frozen down to be used at a later point. But doing this is actually really important. But how often and when should I freeze down my cells? You are often advised to freeze down cells with a low passage number. And this isn't bad advice. The longer you grow cells, the more time they have had to accumulate mutations, which means the cells aren't quite the same as the cells you started with. Now, whilst you might be able to shrug off this piece of information, it has important value because an experiment you do with the cells you have at the beginning of, let's say, your PhD, could be completely different to the results you get using the same cells at the end of your PhD. So how do you get around this issue? Well, as I've drawn out here, one thing you can do is freeze down 10 to 20 or even more than 20 different files, and do this at very early passages when you first start with a new cell line. So when you finish your experiments, you can actually throw those other cells away and just get a file that you froze down months ago, and off you go. So the reason you'd want to do something like this is that the reproducibility of your data and your experiments is really good, well, at least better than what it would be if you started with cells that had gone through multiple passages and gained many different mutations. And so this idea is often referred to as your working cell bank. So for the second tip, cells come in all different shapes and sizes, and some with genetic modifications, such that looking at them under a microscope will not be sufficient for you to distinguish between two different cell lines. So how do you know if the cells that you're working with are actually the cells that you think you're working with? Well, you should somewhat regularly do an STR analysis. And so STR stands for short tandem repeats. And it's a kind of like a forensic detective DNA-based method to determine what cells you're actually working with. Another thing you should do even more frequently is to check for mycoplasma. So mycoplasma are bacteria that can contaminate your samples, and so mycoplasma rest on the outside surface of cells and they can alter the cell's behaviour, in which case how do you know if you have contaminated cells that your results are what the cell would normally do or due to the mycoplasma affecting the cell's life? So tip 4, and speaking of contamination, one good rule to follow is when in doubt, just do it again. If maybe you touch the surface with your tip, your stripette, just get another one. It might seem wasteful sometimes, but the waste and the cost involved in the long run, when one tip compared to a contaminated set of cells, one is evidently much cheaper and less time consuming. So, with time being the money currency in science, just be more cautious than in other situations. Alright, so my next tip is beware of the bubbles. Now, not those sort of bubbles, but bubbles that you can get from aspirating that little bit too fast. Now, this is honestly much easier said than done, but the key point here is that your cells can get trapped in the bubbles and we don't want to lose our cells. Alright, so another lesson / tip is to remember the fact that everything that you do to your cells is going to act as a selection pressure. So whether that's, during your centrifugation to pellet your cells, whether it's how resistant your cells are to trypsin, how vigorous you are with the aspirating, some cells will be able to tolerate it more than others. In which case, over time, you will effectively be selecting for a subset of the cells that you initially started with. Hence, like similarly to the idea that you shouldn't be working with high passage number cells, is that cells are going to change, they're going to evolve, natural selection in the culture dish. I don't really know what that's called, natural artificial culture selection. Yeah, that's a thing. But it's just something to bear in mind and to keep at the forefront of your mind. And so lastly, to break the myth that cells grow faster at the weekend, this is due to the fact that being at the back of the incubator is better for your cells because they're less affected by the door constantly being opened and closed. So at the weekend, when in theory less people are there, that's up to debate, the doors open less often, in which case your cells, they're going to grow at a faster rate, a relatively fast rate. Yeah. So that concludes my tips for getting cultured in cell culture. So you're now as cultured as I am, hopefully. So let's take off our blue or green coloured coats for tonight. And remember, when in doubt, get the ethanol. And as always, thanks for listening.